Thank you for your nice introduction. And in fact, let me tell you, though my designation is principal scientist, I prefer to tell everybody that I am a student. I am a student of science.
That's, yeah, sir.
Because what I feel that more I am working on the area, more I am learning, and I am feeling that there are more to learn. I don't know whether I can finish while in my service life that everything in a single topic or not. But till I am trying to learn many things. And for the students, let me tell you one thing that I did my PhD in plant pathology, but while working in plant pathology that time, I worked on components of slow blasting resistance rice blast disease. That is a serious disease of rice, number one disease of rice. And that time I worked only statistical modeling of rice blast, how it interact with the crop, and what are the epidemiological factors, what are the environmental factors, and just to make a statistical model and prediction. And that time I never thought that I'll jump to another line where I have no strength, or you can say no grip on the subject. Because after that I joined NRC Plant Biotechnology with Director General of ICAR, Dr. Tilochan Mahapatra, to work on marker assisted selection in Brassica junctia. And after that, I joined one state government institute, the Regional Plant Research Center, situated in Odisha, Bhubaneswar. And there I worked on genetic diversity of horticulture crops and forestry crops, forestries. And then I got an opportunity to work in Israel as a DBT overseas associate. I worked there as a visiting scientist for 1 year. I worked there host pathogen interaction, proteomics and genomics. And then recently I am working in ICR Institute, and there I worked on many aspects of what Madam already has covered, so I don't want to tell. But my message is that for the students, that if you think in the beginning that I cannot do this, I am sure you cannot do this. But if you think that yes, I can, if X is able to do it, why can I not? If X is able to do it, I can do it also. So if you have that zeal and patience, I am sure you will be able to do it. So that is a message for the students from the beginning of my lecture. And the students never be disheartened. And another thing is to remember, I always tell my students that hard work repays with compound interest. Maybe for some time you will be frustrated, but thing is that if you are not recognized by your friends or your near and dear one, you'll be recognized by others. Okay, so don't be disheartened. Give your effort. Try to read as much as possible. Try to learn, and you will definitely win. You will definitely conquer. And Madam, I can start now.
Yes, sir. Yes, please.
Okay. So today's topic is that microbial diversity in sustainable health management of crop. This is a very broad topic because microbial diversity, microbial diversity, microbes is everywhere. If you want to see, you just take a petri dish having nutrient agar or some other media and expose it for 10 to 15 seconds in the air, you will see hundreds of microbes are growing on the media. Okay, so just imagine the diversity. And you will be surprised that even 15% of the diversity we could not explore till date. So what we have done means our laboratory is doing, we are using one fungal species that is known as trichoderma, and that is beneficial as well as harmful also. I discuss in detail. And that one we have used for different purpose, for sustainable management of the crop. Why I am telling sustainable health management of crop? My point is that I to grow a particular crop which is healthy for human being, I am destroying the environment. So do you think this is sustainable? No. Why? Now, because our existence, very existence depends upon the health of the soil, health of the air, health of the environment in a total. Health of the environment also important. Even you can see that if you don't have a healthy neighbor, it's a good neighbor at your home, then you can see that your children are spoiled. Maybe that is a chance. Okay, and then you are not in a peace. That means you are losing mental peace. You are becoming mentally ill. So in that case, I should tell that whatever we are doing, whatever we are thinking, it has to be sustainable and it has to be healthy. Okay. So now why the sustainable management of crop health? This goal of sustainable agriculture is to meet society's food and textile needs in the present without compromising the ability of future generation to meet their own needs. In a recent lecture I attended, one lady from America, she was telling that in another 50 years we are going to be 17 billion population. Just imagine. In India, we are having population at present approximately 1.4 billion. So in future, we are going to cross 17 billion, and the 17 billion means whatever food we are having at present food production, we need just to double that to feed all the people, and the grow the crop. To grow the crop, we are losing huge amount of groundwater, and the need of water, what was there 25 to 30 years ago, it has become 4 times less now. We are getting 4 times less water. Why? Because we are not able to manage it sustainably. We are not able to manage in a healthy way. And sustainable agriculture describes the landscapes that support both agriculture production, biodiversity, and environmental conservation, working in harmony together to improve the livelihoods of the rural communities. Just imagine, because in urban areas, we are able to purchase all the health. Health means how? We are staying inside the AC room, we are able to have water, air purifier, but even then we are not actually healthy. If you can see the present COVID situation, if you go to the village, I see the villagers are better position than us, because when I visited my native place before 1 month, I saw nobody is wearing mask and nobody is ill. But in city area, the people are wearing masks, they are taking all the cares, but even then they are falling the victim of the COVID-19. So what are the steps for sustainable management of crop health? That is management of crops without chemical pesticides and higher harvest even in adverse condition. Then proper management of crop residues. Just remember these 3 points, that chemical pesticides is not only dangerous for human being, but it is dangerous for the entire environment. How? You can see, if you put little bit of pesticides in your pond, all the fishes will die. So you are losing all the fishes. There are many pesticides which are killing the beneficial insects as well as beneficial microbes. Then higher harvest even in adverse condition. What does it mean? Now in present day, you can see the drought condition is prevailing everywhere, either drought or flood. You can see either in Chennai side it is flood, in Rajasthan and other places it is drought. Even in a single state, different districts, there are different type of environments. So you should have higher harvest even in these adverse condition. So your crop or your crop management has to be designed in such a. Then again, if you can remember that before just 1 year or 2 years, the entire Delhi was standstill due to burning of the crop residues in Punjab and Haryana. And it was found by the FAO and WHO that this burning of the crop residues, the suspended particles, they are moving even up to UK. Just imagine. So they are not polluting the surrounding area, but it is polluting a huge area. So just one request to my students, that if you feel anywhere that you are not able to understand, just stop me, I'll answer. Then in modern agriculture practices and environment, is it eco-friendly? No, it is not. Why? Now the practice what we follow is develop soil erosion, means it erodes the soil and the entire soil is washed out, topsoil is washed out and it is going to the water, and by means of water it is going to the sea ultimately. Then sediment transport and deposition downstream, means that sediment is being transported and it is deposited to the rivers, and rivers are blocked, and then as a result we are facing flood. Then on-site pollution from overuse and secondary effects of fertilizer pesticides. If you can remember that Bhopal gas incidents, many people died, and till date many people are suffering due to cyanide leakage. Even recently in Telangana district also it was havoc. That offsite pollution of other ecosystem of soil, water and air. Means when you are applying any pesticides or chemicals to your crop, it is going to the soil, it is killing number of beneficial insects on the topsoil, and then water, it is going to the ultimately water, and it is polluting the water, and also through air it is being inhaled many persons. Yes, please. Many persons, and they are also getting affected. Then modern agriculture is not practices, agricultural practices are not sustainable as it is not environmental friendly. As I told earlier, that what are the problems are that to feed the huge growing population, we are going to have use new lands, and for that we are going for deforestation. Then due to deforestation, we are converting the fertile land to desert. Then the aquifers, means whatever water sources are there, we are degrading those. Then we are accumulating huge number of toxic organic compounds. As we are going for deforestation, we are losing biodiversity, and our agriculture practices for a particular selective crop also is just reason why you are losing biodiversity. For example, if you see, I don't know, you are students, so you are very young, but in our young age, we have been visited different marketplaces as well as different mango orchards, where we have seen various types of mangoes, various types of crops like brinjal. India is the origin of brinjal. So brinjal also, even if you go to the village markets, you will see various shapes, sizes of brinjal. But due to introduction of the BT brinjal and other brinjals which are high yielding, we are preferring to only one or two types of brinjal. So ultimately, those which was diverse brinjal varieties of brinjal species or brinjal available in the market, we are not getting those now. We are losing it, and in the mango also, you will see mainly bygone foli, a typical one type of mango prevailing, particularly in southern India as well as this part of eastern India. Then environmental pollution, as I told, when we are trying to manage the crop density, we are burning it because we have to make our field clean immediately. So we are burning them and we are polluting the environment. So in that case, question will come. Then what is the way out? Should we stop the agriculture? No, we cannot, because we have to feed 7.6 billion people at present, and it is increasing very rapidly. Okay, and all of them need food. So that means we cannot stop agriculture. We have to go with agriculture, because agriculture is the source of food and source of energy. Then we have to also think for preserving our resources. Why? Because I don't know how many of you know that rice is affected with a serious disease known bacterial blight. And that bacterial blight is a very severe in almost all over India. And we have got a bacterial blight resistant gene, which is XA21, that is from wild rice, that is Oryza barthei. Okay. Oryza barthei we got from Oryza barthei XA21, one resistant gene that is working excellently almost all over India and abroad. Okay, that means these wild genotypes are source of many beneficial traits. Okay, so we cannot destroy them, we have to preserve them. Then what is the way out? The way out is that we have to search of alternate way to manage the crop health and manage the crop residues, and huge diversity of microorganisms can help here. Okay, which I was telling. That means we have to find out some alternate way and to manage the crop health as well as crop residue. And there, that microorganism, which are huge in diversity, can help. How? I'm going to tell you. Then this use microorganisms for crop protection against biotic and abiotic stresses, growth promotion and crop disease management are the alternatives. That is my point of saying. Okay. You can see the food grain production and pesticide use in India. You can see with the production of food grains, we are using the more pesticides. Okay, and it's very havoc day by day. I have not given the detailed data. I've just up to 2030 I have projected. This is I have taken from internet. It's not my own data. And then pesticide scenario in 2010-11, that again I took from internet, and you can see here almost all the states are using huge amount of pesticides, and highest pesticide is being used in case of paddy. We think that cotton is in cotton we consume more pesticides, but fact is that highest pesticide is being used in paddy. And then pesticide consumption over the years in India, you can see here over the year it was with introduction of BT cotton, it was reduced in 2001 to 2005-6, but again it is shooting up. Then consumption of chemical pesticide in various states during 2014-15 to 2019-20 in metric ton technical grade. You can see here the highest is being used in Maharashtra and followed by Uttar Pradesh. And other states are also no less. Okay, but there are few states we are using very less, and some states like Sikkim and other northeastern states, they have converted to organic agriculture, so they are not using at all any pesticide. Then, but fortunately in India, we are the using lowest amount of pesticide. That is a very good thing we are doing. If you think in Taiwan, Japan, Holland, South Korea, France, or USA, in comparison, we are using very less pesticides. Okay, and these pesticides are polluting the environment, as well as you can see here they are polluting the animal husbandry and public health programs also. Then it is estimated that less than 0.1% of an applied pesticide reaches the target pest. Just imagine. That means if you use 100 kg of pesticide, that out of 100 kg, how much it is reaching to the A? It is reaching only 0.1 gram. So sorry, 1 gram, and 999 gram is not being used. It is being used as polluting the environment, polluting the soil, air, and water, or nearby vegetation. Just imagine. Then, in this case, how we can use microorganism for sustainable management of crop? First thing is that we can use biocontrol agents for management of disease and insect. You know biocontrol? Any student? Any student, please? Hello?
Student, please discuss with sir. Whatever he ask you, please reply.
Any student is there who knows what is biocontrol?
So they are mostly from biotechnology, some are from microbiology, and there are one or two other subjects.
Okay, then let
Yes,
me.
sir, biocontrol.
Yeah.
So we read this in class 11 or 12, that biocontrol, or it is also known as biological control, it is controlling pesticides using other organisms, controlling pesticides or something else.
No, not
controlling
pesticide.
pest, insects, weeds.
Not pesticide, like controlling pest.
Controlling pest. Controlling pest. Instead of pesticide, we are using biological pest.
Yes.
That is, we are depending on other organisms for controlling the insects or the weeds.
Exactly. Because instead of chemical pesticides, we are using some living organism to kill another living organism.
Because yesterday only we had a lecture on, biocontrol agent, fungicide by Dr. Ramesh Chand.
Oh, yeah, I know him.
Yeah.
He's from BHU.
Yeah, yes, sir.
Okay. Then for enhancement of plant growth, hence reduction of chemical fertilizer, management of abiotic stresses like drought, then your salinity as well as flood, then eco-friendly management of crop residues. In those cases, our microbial diversity can be helpful. And here in our lab, we are working with a fungal species, that is Trichoderma, for sustainable management of crop. I am going to show you one by one example, so that you will be able to know how the how we can use our trichoderma diversity for management of crop health. Then question will come, why trichoderma diversity? Reason is that species of the genus Trichoderma is one of the most useful microorganisms having huge impact on human welfare. This Trichoderma is not only being used as biocontrol agent, but also it is being used for production of cellulose by degrading cellulose from for production of biofuel by degrading cellulose from wood and other crop residues, as well as it is number of beneficial enzymes are being produced by this trichoderma. So it is being used for industrial production of number of enzymes. Then these trichoderma species have had major impact on agriculture and known to enhance growth and control of soil-borne pathogens. Okay, we have done some works on that. And these fungi not only protect plants by killing other fungi and certain nematodes, but induce resistance. Means what? That if it is not able to kill the pathogen, what it is doing, it is inducing resistance against the plant pathogen. So exactly what we are thinking about development of vaccine, what vaccine is doing. Okay, it is inducing resistance against plant pathogens, and then it also imparts abiotic stress tolerance and improve plant growth and vigor. Then stabilize the plant nutrients, means it helps the plant to absorb more nutrient from the soil, and then it also bioremediate heavy metals and environmental pollutants from the soil as well as environment. Then trichoderma play important roles in biodegradation and recycling of complex polymers like lignocellulosic wastage and chitin. So using this property, what we can do, we can decompose our crop residues and use them as manure, as fertilizer, or you can say manure rather, or we can add organic carbon to the soil. Then there are a number of secondary metabolites identified like viridin analog, wortmannin, trichostein, and peptibiotics are potential anti-cancer and antimicrobial drugs. Just imagine. And research is going on that. You see, from a single genus, Trichoderma, you are getting many benefits. Okay, even they are being used as a potential anti-cancer drug and antimicrobial drugs. You know that at present there that antibiotic resistance is a serious problem, and if we can use an alternative of bacterial resistant drug, bacterial isolates which are resistant to different drugs, okay, the antibiotics. So if we avoid that and use product of this trichoderma for as a antibacterial property, so then that will reduce the use of antibiotics, and then we can have better health. Then ability of these fungi to sense, invade, and destroy other fungi have been the major driving forces behind their commercial success as biofungicide. Just imagine that whatever biofungicides have been registered so far, 60% of them are based on the trichoderma. Just imagine. And you'll be surprised to know that in India alone, there are 250 commercial formulations being sold. Okay, and these entire commercial formulation, they are based on a single strain, that is Trichoderma viridi, recently classified as Trichoderma aspergilloides by us. And this Trichoderma viridi was developed by Tamil Nadu Agriculture University. Then this is a pictorial depiction that how trichoderma is helpful in human welfare, or how it impact, what is the impact of human welfare. And you can see that they are not only beneficial for the human being, but also they are having hazard to the human being. Okay, for example, remember this point, any trichoderma which grows better beyond 35 degrees Celsius, they are usually human pathogen. Remember this point. Then As trichoderma has had a major impact on human welfare, recent genome sequencing projects have targeted 7 species. So I'm not telling the name, and not only 7, actually it is 8 species, because we have recently added another one from our lab. Then 200 trichoderma species have been recognized on the basis of molecular phylogeny. The systematic position of trichoderma is as follows. Its kingdom is fungi, division is Ascomycota, subdivision is Pegizomycotina, and class Sordiari Mycetes, order Hypocrealis, family Hypocreaceae. Then in our lab, we have used these trichoderma species. Okay, you just see that CRRI T12, NRRI 28, there are different species of trichoderma, and we have used these here. Actually, we have 5 species, and these 5 species we have used for 5 different works, and this has been published in Microbiological Research in 2018. Then trichoderma as biocontrol of disease. I was telling what is biocontrol. I asked one student, and she answered beautifully. But here you can see the definition. The biological control refers to the purposeful utilization of introduced or resident living organisms other than disease-resistant host plant to suppress the activities and populations of one or more plant pathogen. Means these biological control are purposefully very utilizing or introducing another living organism to have control over plant pathogens. Either plant pathogen may be one or maybe more. Then why do we need biological control? Biological control is needed because we need to replace chemical pesticides. In recently, Central Insecticide Board has banned a number of fungicides, and out of all those fungicides, there are few fungicides which are regularly being used, for seed treatment. So in that case, if we don't have these fungicide, then how to treat these? If we don't treat the seeds, we are exposing our crop to different soil-borne and seed-borne diseases. So for that, we had to need biocontrol agents. Then what should be the criteria of biocontrol agents? It should be non-toxic to human or animal, non-polluting, and should not have effect on other species. That is one or few species. That means it should be environment friendly. Then present scenario, can biocontrol agent replace chemical pesticides? At present scenario, it cannot, because you know that if we take homeopathic medicine sometimes do wonder, okay, but it takes time to cure. But when a patient is very serious, that time you have to go for to apply allopathy. Okay, but here what we can do, we cannot replace chemical pesticide, but we can reduce the use of chemical pesticides. How? For example, recently our trichoderma agents are being tested all throughout India and they have reported that instead of 3 times use of pesticide, they are using 1 time pesticide use and 2 times biocontrol agent. Okay, and you just imagine a single pesticide at the rate of 2 gram per liter, and for per hectare we need 500 liters of solution. That means 500 into 2 gram or 1 kg. 1 kg for 1 hectare. Just imagine, and few million hectares we are using for cultivation. Okay, and you just imagine how much amount of pesticide is being reduced if we are reducing the pesticide use for 2 times. Instead of 3 times, we are using 1 time only. That means 2 times we are reducing, then it is being replaced, or you can say we are giving a chemical pesticide immediately, and then we are using biocontrol agent. Okay. Then where from to get the biocontrol agents? These biocontrol agents are better if we collect them locally, because all the microorganisms, they are adapted to a certain environment. Okay, so if you induce on biocontrol agent from outside, they take some time to adapt to the present condition, environmental condition. For example, I don't know how many of you know that Gossypium hirsutum is a cotton. To grow cotton in India, that is known as American cotton actually. That cotton in India, it took years just to adapt in the Indian soil. And ultimately they are adapted to Indian condition, and now we are growing American cotton. Then you can see here, this is one review article, it published recently, and you can see that how trichodoma is having impact on rhizosphere. This is the plant, and you can see it is improved 5 physiochemical processes, better absorption of mineral nutrients, better reproductive potential and yield, enhanced morphological growth, and the regulation of phytohormones levels. Means it is the mechanism how it is being done, and it is the effect. Okay, so this is the way how trichodoma is interacting with different crops in different in their different morphological morphochemical characters. Then what is the mechanism of trichodoma as biological control agent? One is mycoparasitism. Mycoparasitism means what is it is myco. This is a myco. Actually, myco means fungus. So it is a myco. So it is having parasitism to other fungi. Okay, that's why it is mycoparasitism. That means this trichodoma, it penetrates into pathogen mycelia by degradation of its cell wall. I'll show one picture. Then antibiosis, it secretes number of antibiotics. Then that antibiotic, it prevents the growth of other pathogens. Then competition for nutrients or space, it competes with other fungus for nutrients as well as for their space. Okay, means where for the locality where they are growing. And as trichodoma is very fast growing, so trichodoma mostly wins. Then induced resistance, it secretes some chemicals or it induces some resistant genes in plant host, induce the activity of some resistant gene in the host, and the resistance is induced and it becomes resistant. Then inactivation of the pathogen's enzyme. The pathogen, what they do, they actually secrete some enzyme to kill the host. And this trichodoma, they secrete another enzyme to inactivate the pathogen enzymes. So by that way, it protects the plants from the pathogen. You can see here, which I was telling, you can see here, this is electron microscope photo. Here, they inserted mycelia in the, this is a pathogen mycelia, and they inserted the mycelia and they have taken all the nutrients. You can see here by this way coiling, and this is photo taken by us, one of my student. You can see this is Helminthiasporium oryzae mycelia, and this is trichodoma. It is supercoiling, and by supercoiling, it is inserting its mycelia into the your pathogen mycelia and taking all the nutrients. And by that way, the pathogen is being killed. Now, while working in cotton, we identified a new pathogen for the cotton. Okay, and then we wanted to have control over that. So we identified different trichodoma species we isolated, and you can see here, this is a sick soil, means it is full with pathogen, but no trichodoma treatment in the seed. And this is sick soil, full with soil is mixed with pathogen, and then seeds are treated with trichodoma. You can see the plants stand. Here only 2 plants are standing out of 10, and here you can see 7 plants are standing out of 10. Just imagine. Okay. Then this has been published in 3 biotic journal. Then in rice, how it can be managed. So what happened till date, whatever trichodoma has been reported so far, all are isolated from soil only. Okay, and for the first time, we have isolated this trichodoma from tree bark. It is growing in the tree bark, and then we have isolated them. And then we have made single spore culture for purification, and you can see they are morphologically, they are totally different. All are totally different. That means they belong to different species. And you can see this is a microscopic photograph. This is the typical characteristics of trichoderma. Finger-like appendages are there, conidiophore. These are not conidiophore, these are known as conidiophore. You can see here, it is just like finger, and above that conidia is there. This is conidia, round shaped. Okay, and this is a very typical character of trichoderma. So if you want to identify trichoderma under microscope, this is the typical character. You have to see finger-like appendages where the just like this, and here all the conidia are there. These are conidiophore and these are conidia. And then we use molecular characters characterization for trichoderma identification. We use 3 different markers. One is ITS, internal transcript segment, and then RNA polymerase, and then the translation elongation factor or TF. Then we use the phylogram for identification of the trichoderma. You can see here we have identified them as different species, which I have shown earlier in the tabular form. And then we have used different, we wanted to see that whether these trichoderma can really act as a biocontrol agent or not. Okay. So for that, what we did, we have taken 2 serious soil-borne pathogens. One is Rhizoctonia solani, another is Sclerotium oryzae. And then this is known as dual culture. Here actually what we do, as you are undergraduate students, so for you only I am telling this in detail. Here actually what we do, we grow in a plate. One side is trichoderma, another side is pathogen. Okay. Here also you can see here, this is pathogen, this is trichoderma. Okay. And then we grow them. And after some time, we see whether pathogen growth is inhibited or not. You can see here, pathogen growth is totally inhibited. That means this trichoderma is able to inhibit the pathogen growth. And in some cases, you can see here, the trichoderma is overgrowing the pathogen. Okay, the trichoderma is overgrowing the pathogen. That means the pathogen is completely killed. Okay, but some trichoderma are not able to kill the pathogen. Rather, pathogen is overgrowing the trichoderma. So we have rejected this, at least for this purpose. Okay, and this growth inhibition we have depicted in tabular form as well as in graphical form. Then we have identified them as a biocontrol agent. Okay, but question is that they should not be harmful to the crop. Okay, for that what we did, we treated them with trichoderma and compared them to see what is the germination time in rice varieties. Okay, so that we will tell that it does not have any adverse effect on the rice or our target crop. And you can see here, this is Corona and Sahuvagidan. The time taken in control is much higher than time taken in the treated. They are treated with trichoderma, different trichoderma species. Okay, that means it is not having any adverse effect, rather it is having beneficial effect. How? Now it is reducing the germination time. Here it is taking up to 4 days, and here within 3 days all are germinated for Sahuvagidan. And Corona, it is taking around 2.5 days, but in other cases it is almost within 1 day they are being all getting germinated. Okay, then when we treated with the trichoderma, the plants, sorry. We treated the plants with trichoderma. We wanted to see which is the best. Then we have different criteria. Out of that, one is chlorophyll content. And in CRRI T2, one strain, it is, you can see here, in all the cases, the chlorophyll content is much higher. Okay, that means it is helping the plant to remain green for a longer time. Okay, so that we have more photosynthetic efficiency. Means more photosynthetic efficiency means we will have more harvest, more yield. Then, as I told, the nutrient absorption capacity, it increases. You can see here that it increases the total uptake of NPK by plants in rice varieties. You can see here, this is control is much less than that of our trichoderma species. That is, we have selected these T2 and T3 for our further study. And you can see here in T2 that our NPK content is much higher in comparison with the control. See here, it's almost double, just almost double. See here. Okay. And then what's happened to the effect of on the crops about the disease incidence? So is it working as a biocontrol agent or not? So for that, what we did, we treated the seeds with trichoderma and grown in the field, and then we saw the incidence of the diseases. Okay, and you can see here when we treated the CRI T2 and T3, if this is control, you see control, the incidence of disease is almost 80%. Okay, whereas for in case of seed blight, almost 80%, and in case of seed blight in corona, it is almost 40%. Okay, similar case is in brown spot, and you can see here that incidence is much less when we treat with our trichoderma species in comparison with the control. Then this is a field view, this is control, and these are treated with trichoderma. You see the plant canopy, you can see the canopy is here and canopy here. It is so dense, you can see it's so dense. This is control, no, not treated with trichoderma, but these are treated with trichoderma. You see the colony canopy of the plant, it is so dense. That means it is having more number of tillers, much better plant growth, and as I told, crop health management. So it is able to manage the crop health properly. Then ultimately, whatever you do, if you lose the yield, not a single farmer is going to adopt your technology. So what we did, we have tested the yield also, and you can see here, this is the yield in control. This is red one is corona variety one, highly susceptible, and this blue color one is Sohavagidan, which is highly popular here among the farmers. It is a drought tolerant variety, and you can see here much higher yield we are getting. We have realized using trichoderma as seed treatment. Then that quantification of endochitinase 42 gene in trichoderma treated paddy soil means what we want to tell, that is our trichoderma is working, or is the trichoderma which is available in the soil is working for us, and we are miscalculating the efficiency of our trichoderma. For that, what we did, we have isolated DNA, soil DNA from control as well as treated, and you can see treated, the endochitinase gene, presence of endochitinase gene is much higher than that of control. That means when we treated, it is really having beneficial effect on the crop, and it is growing in a huge amount. And ultimately, this is real-time PCR data. And then we have filed a patent on this. We have also published a paper. Now, we have also, now question is that in our laboratory we have tested, in the farm we have tested, but is it all? Because as I told, it has to go to the farmers, and farmers have to realize that yes, they are benefited, otherwise they won't use it, they will throw it. Okay, many popular varieties have come, but they are vanished. Okay, so what we did, we tested in the field, and in field also we get almost 20% higher yield. Okay, then you can see this is a farmer, this is control, and this is treated. This is another farmer, this is our director. Our director also went to visit its effect in the farmer's field, and he was very happy to see it. And you can see this is the farmer, he used our trichoderma, and it was excellent result was there, he was very happy. And this is again black gram, this is the farmer, you can see this is treated and this is untreated. At the end, I'll show the slides, video, that how treated untreated looks like. And then this is a research study, has been done by MS Swaminathan Research Foundation in Koraput. This is Niger plant. You can see here, this is not treated with trichoderma. You see the plant stand, and you see the plant stand here, it is treated with trichoderma. Then, I think now we can have 5-minute break, madam.
No, sir. You can continue. We can end it at 12:30. So.
Okay. Now by 12:30, I think I will be able to finish.
Yeah. Okay.
Okay. So then second point comes, earlier point, what we got, the proper health management of the crop, means biocontrol of the plant as well as growth promotion of the plant, proper growth of the plant without having any effect on the environment. Second point is now that, as I told, that to reduce the pollution by managing the crop residue properly. Okay, so this point I am coming. Rice straw, you know that almost whatever crop we are harvesting in rice, if we harvest 40 kg of rice, and then we are also harvesting 60 kg of rice straw. Okay, and which become a menace in Punjab and Haryana, where they want to grow wheat immediately within 25 to 30 days. Their field has to be ready. So what they do, they use harvester to harvest it, and then they get the paddy, and then their crop remains in the field itself, their straw. So what they do, they simply burn them, and by burning them, you see the background smoke. By burning them, they are polluting the environment, and many people, they suffer from different diseases. And you can see the India produces 106.19 billion tons of paddy from 45 million hectares area with roughly 130 million tons of straw. Just imagine. This rice straw compost can increase crop yield by 4 to 9% when applied as mulch to help it maintain soil fertility. The compost contains 1.7 to 2.1% of nitrogen. I'll show in detail later on. And Dr. O.P. Rupela, who worked as a scientist in microbiology at the International Crop Research Institute for the semi-arid tropics, Hyderabad, he told that rice straw burning leads to nutrient loss from the soil, which is equivalent to $18 million. Just imagine. Then what are the ill effects of stubble burning? That causes massive air pollution by releasing smog laden with harmful gases such as sulfur dioxide, nitric oxide, and carbon dioxide. It depletes nutritional value and fertility of soil, causes respiratory disorder and other health hazards. It becomes a cause of road accidents due to reduced visibility, irritation in the eyes, which may lead to blindness. Then here, Trichoderma species can have a nice effect, which not only decompose the crop residue, but also that decomposed straw can be used in the to increase the fertility of the soil, and also it will control the soil and seedborne pathogens of crops. Okay, so for that we wanted to characterize our Trichoderma species, and you can see here we have characterizes for there is cellulase, xylanase, and lackase producing activity in qualitative analysis as well as quantitative analysis. And you can see here, this is quantitatively we have analyzed. This is another species, NRRI26, that was NRRI2, this is 26. And you can see here that after 60 days of decomposition, this is the straw where no Trichoderma treatment has been given. And you can see this is a straw where Trichoderma treatment has been given. And then enzymatic index of exhibiting cellulose, xylase, and lackase activity of Trichoderma isolates. As I told, NRRI26 is the highest one. So we have selected this one. Then in this rice straw we have used in the soil, and we have seen that all the characteristics like gravimetric weight loss, then gravimetric weight loss of decomposed rice straw, everything, total nitrogen in percentage, total phosphate, everything, this is much higher in comparison with the control. Okay. Then this you can see here, it is very clear that this is a control, and these are grown in straw decomposed, Trichoderma assisted decomposed rice straw. And these are different meetings we have conducted, forget it. Now, that point is over. That means we are able to manage the residue properly. But again, we have to, we are working till now to reduce the time, because the farmers need, it has to be decomposed within 30 days. So we are trying to develop a consortium so that in consortium it can be decomposed within 30 days. Let us see what will happen. And this one is Trichoderma for abiotic stress. Okay, this is another species. You can see here, this is control, and these are different Trichoderma species. And you can see here, all the plants are surviving. All the plants are surviving and green even after 7 days of water withdrawal. Just imagine, 7 days of water withdrawal, they are still surviving. Okay, but here you can see in the control, they are totally finished. Then this one again, this is another isolate. You can see here, they are also again remaining surviving after 7 days of water withdrawal. And then you can see this is a tabular form we have presented. You can see this T4 and T13 and T11, they are excellent for the drought, inducing drought tolerance in the ice. And this is again the percentage of germination as well as how much survival after 7 days of water withdrawal. And what was the mechanism? To study that, what we did, we grow them in different one approximately 15 centimeter diameter pipe, one seedling, and to see that pipe length was 1 meter. And you can see here, this is a control, this is a drought tolerant. That means it's normally it is having longer root, and this is drought susceptible. It is having very less root, very short root, so it is highly susceptible to drought. And when we treated with trichoderma, you see the root length. Just imagine the root length here as well as here. So even we stopped water for even 15 days, the plants survive because they are able to have penetrate the roots into much deeper of the soil. Okay, and then this is average root length in IR20, that is drought susceptible, and this is CR143, that is drought resistant. And interestingly, we have seen better effect in drought susceptible than that of drought tolerant, because drought tolerant is already adapted to have much longer root length in comparison with the drought susceptible one. Then this one we grown in our field, and under field condition even after 25 days they remain green. That means this trichoderma treatment, if we can use for the farmers, even if we can avoid the 7 days of drought, that is huge achievement for rice. Just imagine, because you know that in Indian scenario that rainy is a very irregular, the rain is very irregular. It is heavy rain for 10 days, then stop for 7 days or 8 days or 9 days or 10 days, then again heavy rain. So these 7 days drought spell, it kills most of the plants. So if we can avoid this by using this trichoderma treatment, then I think this is a wonderful achievement. We are going to publish this in a journal. We are writing that paper, and also we are trying to have a project from DBT to make it in the to bring it in the farmers field and popular. And then consumption of biopesticide formulation in various states. Out of all those things, the scenario is very poor in India. Till date, we are depending on chemical pesticides. But fortunately, in a recent trend, the use of biopesticides is increasing. In Madhya Pradesh and Maharashtra, it is much increased. Tamil Nadu, it is a better one. And Uttarakhand, West Bengal, it is better. And as I told, the number of states, particularly northeastern state, they are not using any chemicals, so they are using huge amount of biopesticide. Then regarding for you, as you are all are students, so for students there are many avenues of trichoderma. As I told earlier, there are 7 species have been sequenced, but we have added another one, so it will be 8. We have sequenced our newly found Trichoderma erinaceum, and then these are the different criteria of sequencing. I'm not going in detail because you are undergraduate students, but if you want to know in details, you can write to me anytime. I am available anytime. I have contact number with madam. If you want, I can share with you also. And these are different enzyme code distribution. These are Venn diagram. This is different types of proteins, how their function are interrelated. It is shown here. And they are different distribution of their functional characteristics. That is biological processes, then molecular function and cellular component. These are different activities which are distributed in the genes. Then you can see here that these functions, how they are functioning in different ways. That is organismal systems, that is environmental adaptation, aging, etc. These many are there, and the environmental information processing. You can see highest is up to 300. That is for translation, and transcription factor are how many? It is almost 120 transcriptional factors are there. Then these are useful genes which have been identified in Trichoderma species. And this again we took from on farmers field. And then in conclusion, I can tell that From all the observation, it was evident that the huge diversity of trichorama species are not only useful for biotic stress management of crop, but if you use judicially, it can be helpful for holistic management of crop plants, which includes management of biotic and abiotic stresses, promotion of growth, and ultimately increase in yield, as well as residual crop management. Now, as your students, then what are the avenues for the students? First thing, research. In research, you know that as I told, only 15 to 20% microbes we are able to identify till date. So almost more than 80% to more, maybe more also, are not identified properly, and we have not characterized them. We don't know what are their effects, whether they are beneficial or not. So you can focus on that. Then, as I told, even yesterday I was listening a lecture from one lady from America. She was telling that in rice, submersion tolerance one sub-1 A gene is there. Okay, but there are problems. The submersion tolerance means when they are submerged in water, they survive. Then when the water recedes, the plants they survive again and they grow and they give the yield. Whereas those who plants which don't have submersion tolerance, they all die. But in her experiments, she saw that IR64 on variety with sub-1 gene, they all survived after submergence. But IR64 which don't have submergent tolerance gene, the sub-1 gene, there are also few plants that survived. Okay, so there is a question that how it is possible. Then she told that we have identified only sub-1 few genes, sub-1A, sub-1 gene like that, but there may be some minor QTLs which may be responsible for submergent tolerance, which we have to yet identify. Just imagine, I am talking about rice. Rice is the most studied crop so far, rice and wheat. So if rice and wheat it is situation like that, the microbes where we have not studied so far so much. So you just imagine the opportunity available there. In a recent another webinar from Israel, Dr. Vitero from Italy, he was telling that in rice there is no microorganism reported so far, which is beneficial for rice growth or biocontrol agent. Then I told him that Dr. Sir, we have one, and then he asked me to send my reprints. I sent the reprints immediately, and he was highly impressed, and he congratulated me, and he is interested to work with us also. Then identification of secondary metabolites responsible for beneficial effects. If you identify the secondary metabolites, so you need not grow the microorganism. You can simply synthesize these secondary metabolites, use them for the plant. Okay. Then if you want that you want to go to business, you are not interested for research, you can do that. You can identify beneficial microbes, make different formulation, sell them to the stakeholders like farmers and industries, and earn money. Okay, and then service to the nature kind. You can serve the nature. How? Now, as I told, if you can use reduce the crop residue burning, if you can reduce the use of chemical as fertilizer or pesticides, you are helping the nature to sustain its health. You are helping the mankind to have a healthy air, healthy water, as well as healthy living. So I have 2 points to say for the students. Sorry. You have to dream before your dreams come true. That is told by great APJ Abdul Kalam. And our great Rabindranath Tagore, he told that he cannot cross the sea merely by standing and staring at the water. Okay, is for the students, it is, you should remember this thing. And let me tell you, my strength is my students. These are my group, they are my students working with me, and this person is my technical officer, and these are my assistant, lab assistant, and other all are my students. Some are postdocs, some are PhD students, and they are actually my workforce. So my dear students, maybe in future I may get one of you as my PhD students in my lab also. Okay, so please focus on your work, read properly, and don't hesitate to ask any question to me. And I don't know whether I am able to convince you that microbial diversity can be used for human benefit or not, but I have tried my best. I am ready to any question, madam.
Yes, so students, you are free to ask me.
Hello, sir. Good morning, sir.
Good morning. Good morning.
Sir, my question is, trichoderma, sir, is a fungus. So it is used for soil treatment for suppression of various diseases, or sir, treatment for the treatment in the plant, sir, for the various kind of the disease, sir. I mean, sir, treating in the soil or plant, sir.
Actually, it can be done both way. What we are doing actually, we are giving seed treatment, okay, so that we can divide soil and seedborne pathogens, and we have one advantage, that is our trichoderma species is endophytic. You know endophytic trichoderma, they enter through roots inside the plant, okay, and then they resides inside the plant and give strength to the plant to fight against different adverse conditions. And also it promotes the growth and ultimately you get higher yield. Okay, but we use as a foliar spray also. We use for soil application also with FYM. So anything can be done, no problem, absolutely.
Okay, thank you, sir.
So any more?
Thank you, sir.
Thank you, thank you. That means I am not able to make you understand properly.
Student, please discuss because we have time and you have this opportunity to directly interact with scientists. So it's, you know,
Excuse
you're
me,
not
sir.
going to have this opportunity again. Yes, Laksh, please.
Yeah. Sir, I had this one small doubt related to this.
Yes, please.
Yeah. Can we do so that is this kind of this kind of the functioning is applicable to aquatic plants as well?
As I told that our trichoderma is endophytic.
Yes, sir.
So for endophytes, you can work in aquatic plant also. And you know that in aquatic plant also there are many microorganisms. I'm not talking
Yes,
about trichoderma. And those are microorganisms, if you can identify and if you work them and if you use them, definitely, why not?
yes, sir. Thank you, sir.
You can ask any question, no problem.
So one question in chat box that what if we discover any microorganism or any herb which can be useful by then how should we proceed with research as undergraduate student?
For undergraduate students, I don't know whether you have the opportunity to do the research or not, but in India, I'm telling you in India, but in abroad, when I was in Israel, one girl was working with me. She was doing BTech. It's very funny in India that if you do BTech in mechanical engineering or electrical engineering, okay, you have to find some circuit or some machine, and that girl was working on biocontrol agents with me as a graduate student. She was doing her thesis, okay, and she did excellent work. Really, it's a really splendid work she did. And for you, if you have the opportunity to work as for research as an undergraduate, you can do it. You simply the steps I told you, that you isolate the microorganism, test for the beneficial effect in the lab. After that, you see that they should not have adverse effect on the crop or your target organism. And then further you want to do anything, you can see the what are the genes responsible for that, what are the secondary metabolites responsible for that, and how it is inducing resistance, how it is promoting the growth by way of secretion of hormone, by way of some secondary metabolites. Everything you can do, absolutely no problem.
Sir, there's one more question related to the trichoderma.
Hmm.
Sir, as you said that it penetrates to the organism, the plant body, it penetrates inside. So, does it not happen that the immune system responds back?
That's what I told you, na, induced resistance.
Okay, sir.
Yeah, that I told, na, that is, you can see what I can say, non-host resistance, that is you can say. Non-host resistance means that what happens, the trichoderma is not a pathogen to rice. Okay, I'm telling you. So in that, but that, pathogens triggered your resistance or whatever you say, molecular mechanism. So in that case, what happens, whenever that trichoderma inside, what is introduced inside the plant, then plant senses that some foreign body has come inside it, just like immune response in our body.
Yes, sir.
So
Yes, sir.
then plant, what it does, it induces some resistant genes, or some resistant genes, they are expressed, overexpressed. By that way, they are, as trichoderma is not harmful, so it does not have any effect on the trichoderma, but it have indirect effect on the pathogens. So it becomes beneficial for the plant.
In a way, it increases the inducive resistance for plants against pathogens.
Yeah.
Yes, please. Who wants to ask? So, sir, yes, please. Who is that?
Ma'am, Visalachi here.
Yes, please. Go ahead.
Sir, why these biocontrol agents are not serving as substitute for chemical pesticides and all?
As I told, I have given one example to you, if you can remember, that is homeopathic medicine and allopathic medicine. Okay. So what happens, till date, we are not able to have a good biocontrol agent which can completely replace the chemical pesticides. But as I told, that say seed treatment we have already replaced with our trichoderma or our biocontrol agent. Okay, that is a huge achievement. We are reducing the use of chemicals, but we are still not in that stage that we can completely replace the biocontrol agents. We are trying that. Let us see.
Sir, also one more thing, sir.
Yeah.
Is there any way that, as we know that in chemical pesticides, there are other effects on the plants as well? It not temporarily, but with the time, it damages the plant in some ways, or the fertility of the soil gets deteriorated from by time being as the time passes. So, is there any kind of a negative effect for trichoderma as well, sir?
We have studied, but till date we have not received anything. But remember, there are some trichoderma, okay, that they produce one gliotoxin, one toxic material. If it is producing gliotoxin, then it is harmful for human being. And then I told while delivering my talk that any trichoderma which is growing more than 35 degrees Celsius, they are actually pathogen to human being. Okay, and regarding the effect on the soil, we have studied so far. We have not got any result, any adverse effect. So, but we are studying now. We are going to do it metagenomics. That is one way of finding the effect on the soil microflora. So there also, if we get some adverse effect, then we have to tell yes, it is harmful for the soil. But other chemical and biochemical mechanisms, whatever we have studied so far, we did not get any adverse effect.
So I have heard about that trichoderma. Some species of trichoderma produces aflatoxin.
No, 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 it is penicillium. Sorry, aspergillus. It is aspergillus. Aflatoxin is aspergillus flavus is one fungus is there. If the toxin is produced by that, aflatoxin is very dangerous for human being. So it is aspergillus, not trichoderma.
So, sir, I had the same question that what if we are using any sort of this biocontrol agent, and what is the effect on human being while consuming that particular grain or something? So, is this study is required? Have you studied that?
Yeah, madam. There are many studies have been already done, and in different fruit crops and other vegetables also. And till date, they have not they did not find any harmful effect. In fact, for any organism to be treated as biopesticides, there is one guideline,
Right,
and that guideline is that you have to go for toxicity study first.
right,
It does not have any adverse effect on cattle. It does not have any adverse effect on human being. It does not have any adverse effect on any other beneficial organism. And there are many other criteria also. If it fulfill all those criteria only, then it will be known as biocontrol agent.
right, sir.
Like Pseudomonas aeruginosa, you might be knowing Pseudomonas aeruginosa, one is there bacteria. Many people are now reporting them as biocontrol agent, but that is a pathogen to human being.
Yeah. So, sir, yesterday we had discussion with Professor Ramesh Chand,
Yeah.
and he personally I asked him because I have also wrote one proposal on biocontrol agent to DBT that is by isolating organism from marine environment. But he tried to convince that instead of focusing on organisms, we should focus on bioactive compounds from the organisms. So, what is your view on that, sir?
Madam, actually, advantage is that if you have bioactive compound, which I was telling in my year, that's true for new avenues for students. If you really have a secondary metabolite identified, and then if you really find that what is the active compound responsible for that, then for industrialization, it is very easy. Okay, you can use many in a very limited area with machines, but here, if you really want to manage the biocontrol agent, the problem is that these biocontrol agents, you grow more and more, sometimes they lose their activity.
Right.
So if they lose their activities, then what will happen? You are simply cheating the farmers or the stakeholders. Because you are thinking that it is working, farmers are thinking it is working, but actually it is not working. They have lost their activity. Like I'm telling you, I am also working on a bacterial blight pathogens of rice. In bacterial blight, if we maintain regularly in the lab, after 12 to 15 cultures, they don't create disease in the rice. That means they are losing their virulence. And these biocontrol agents also that they work also in the similar mechanism because they are virulent to the pathogen. They infect the pathogen because of their virulence to the pathogen. So if they lose virulence, that will be simply you are using normal talcum powder.
Right, sir. And when you are using the live microorganism on the field, then definitely to control the environmental factors and geographical conditions, it affects, right, sir?
Yeah, that is true. Another problem is there, madam. Selection pressure. Because when you are using a particular single, I am working on that now. When you are using a particular microorganism in a huge amount in the soil, then what will happen? As I told, they will fight for nutrient and space. Okay.
Right.
Whoever is growing faster and much virulent, they will grow, and the other microorganisms they will simply be suppressed. So that also you have to think that whether soil microflora are being maintained properly or not.
Right, sir. So students, if you have anything now to discuss or anything you want to ask, apart from this topic, you can utilize the expertise of Dr. Arup.
If they want, madam, they can contact me anytime also, no problem.
Yes, sir, I'll give the email ID. No problem.
Yeah.
Since they are undergraduate student, they are learning different topics in this session. Otherwise, they are from different backgrounds. So
Yeah, that's what I.
Yes, please.
Sir, I wanted to ask one more thing, sir. You said that certain species of trichoderma are harmful to human beings as well, sir. So, is there any way that we can do something related to it?
Yes, it can be done, but thing is that why do you want to do that? Because
I mean, in a way that, sir, the properties does not change as such, and the harmful effects onto the humans decreases, sir.
yeah, it can be. Why I'm telling you that now marvelous biotechnological tool has come, that a CRISPR-Cas9. You have heard that CRISPR-Cas9?
No, sir. I'm sorry.
That is genome editing tool. You simply edit that particular gene, which is secreting, which is responsible for secretion of that harmful toxic substance. Okay,
Sir.
just edit that gene so that that gene will be non-functional and it will stop producing that toxic material, and then it won't be harmful to the human being. And that tool is known as CRISPR-Cas9.
Thank you, sir.
So I think there are no more questions. Hello, students, can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay, so I guess you don't have question now. And if you have any, you can call me, or you can drop down the message, or you can mail me. I'll give the email ID of Dr. Arup, so you can contact via email with proper query and your purpose, right?
Yes.
Okay, ma'am.
Okay, so sir, thank you so much for your time, and it was really wonderful and informative session, and I am sure the students will benefited from this. They would have detailed study in future, and when invite them for the hands-on training, they have to work on some grassroots practices, and mainly over the year we have documented whatever Honeybee Network has documented grassroots practices. Maximum practices we have for the farmers only, I mean for the agriculture field. So they definitely have to work for this, and it would be beneficial for sure. Thank you so much, sir.
Thank you, madam. Thank you very
Sir,
much for giving me the opportunity.
Sir, I had one last question, sir. Sorry to interrupt.
Yes, please tell me. No problem.
Sir, is there a way that we can use transgenesis for the same?
What I could not understand.
Sir, transgenesis?
For transgenic, for what?
Sir, can we do such that the we can improve, or we can add on some things into the trichoderma for this?
Yes, it can be. It can be, because I am telling you one example. That is Metarhizium. One is there. Fungus is there. That one actually is harmful to the insect, particularly for mosquitoes. Okay, so
Yes,
so what is done? They have incorporated BT gene inside that, and then they have improved their virulence to much more than how much it was there. Okay, by using transgenic approach. Okay, but question is that that Indian system, I don't think they will allow you to do so. Okay. The reason is that we have a very stringent procedure to release transgenic material to the environment.
Yes, sir. Yes, sir. Thank you, sir.
Thank you. Welcome.
Thank you, sir.
Okay.
Thank you. And thank you, madam, very much. Thank you for giving me the opportunity.
Sir, thank you so much, and we always look forward for your support and guidance in such
Yes, madam.
workshop.
Yes, madam. I'll be there always.
Thank you, sir.